Biochemistry 401 Lecture 27. Today we're going to discuss some additional topics in fatty acid oxidation, the catabolism of odd number fatty acids. We're going to talk about the metabolism that happens in peroxisomes, and also about the glyoxylate cycle. We're then going to talk about fatty acid synthesis, the synthesis of saturated fatty acids and unsaturated fatty acids. And finally, we're going to talk about the regulation of fatty acid metabolism, both the regulation of fatty acid degradation and fatty acid synthesis. So let's get started. What about fatty acids with odd numbers of carbons? Well, fatty acid catabolism will proceed as normal, releasing 2 carbons at a time, until the very last round of beta oxidation leaves a 3-carbon fatty acid, propionyl-CoA. This propionyl-CoA will be used to synthesize succinyl-CoA in 3 steps. The first step is a carboxylation. We're going to add a carbon in an ATP-dependent carboxylation using biotin. We end up with D-methylmalonyl-CoA, and we're going to racemize that from D to L-methylmalonyl-CoA. And then finally we're going to rearrange this molecule to make succinyl-CoA. Now this step involves an intramolecular reconfiguration, and this is a big job. And so we're going to use a big cofactor, cobalamin B12. Now this is a process that involves some radical chemistry. And finally, we're going to end up with succinyl-CoA that can enter the TCA cycle. So let's look at each of these individual steps one by one. And we'll start with an overview of what cobalamin is. Cobalamin is a large, complex, ring-structured prosthetic group that contains a central cobalt. This cobalt is organized in the center of a corrin ring that is a lot like the porphyrin rings that we saw previously, in that it is comprised of 4 pyrrole rings. However, these pyrrole rings have different degrees of saturation and different substituents than the porphyrin ring. Additionally, it has another functional group called a benzimidazole ring that helps the prosthetic group to function properly. There is a variable region, shown here by X. This X can involve different functional groups, one of which is a 5' deoxyadenosyl group that is shown here. Now this prosthetic group is used in reactions that involve difficult rearrangements of configuration, and the one that we're going to do to convert methylmalonyl-CoA to succinyl-CoA is pretty radical. This mutase reaction results in an intramolecular rearrangement of methylmalonyl-CoA. Cobalt allows formation of a free radical that's necessary for this reaction. And the reason that this can occur is that the bond between CH2 and cobalt is very tenuous and can be broken easily to yield a radical. This happens through something called a homolytic bond cleavage. And what this means is that one electron stays with the cobalt to reduce it to cobalt 2+, and the other one goes with CH2 to form a 5' deoxyadenosyl radical. This is not the normal heterolytic cleavage, in which both electrons go with one or the other bonding partner, but rather it is a homolytic bond cleavage, in which one electron goes with one bonding partner and the other one stays with the other bonding partner. In this case, we're left with a 5' deoxyadenosyl radical and cobalt that is reduced to 2+. It is the ability of cobalamin to form radicals that allows it to function in this mutase reaction. So now that we have a free radical at the 5' deoxyadenosyl group, what are we going to do with that, and how is this going to affect the formation of succinyl-CoA? Well, the first thing that's going to happen is the radical is going to abstract a hydrogen atom from the methyl group of L-methylmalonyl-CoA to form a CH2 radical there. This is going to spontaneously rearrange such that the carboxyl-CoA is going to migrate to the site where the radical is, and the electron is going to migrate to where the carboxylate coenzyme A was. This is the intramolecular rearrangement, and it happens spontaneously after radical formation. Finally, this radical will abstract a hydrogen atom from 5' deoxyadenosine to form a 5' deoxyadenosine radical again. To reconstitute the functional enzyme, this radical will form a covalent bond to cobalt in the corrin ring. And so from L-methylmalonyl-CoA, we get succinyl-CoA. Cobalamin, vitamin B12, is necessary for this reaction, and it involves radical chemistry. Now, we talked about catabolism of fatty acids occurring in the mitochondrial matrix, but it also can occur in peroxisomes in mammals, and it occurs exclusively in peroxisomes in plants and in yeast, rather than the mitochondria. Now, the peroxisome is bounded by a single membrane, and this organelle is thought to arise from the endoplasmic reticulum. There are many enzymes that are found here that catalyze reactions that result in the production of peroxides, H2O2. In mammals, long-chain fatty acids are catabolized here to form shorter octanoyl fatty acid chains that can then go to the mitochondrion for further degradation. In this slide, we see the first cycle of fatty acid breakdown as it occurs in peroxisomes. It's different from what we see in mitochondria. In this pathway, acyl-CoA dehydrogenase uses FAD to oxidize the acyl group to yield a double bond. In the process, FADH2 is generated. This FADH2 passes its electrons off to molecular oxygen to form peroxide. And then an enzyme called catalase can actually break down this peroxide to form water and 1/2 O2. The fatty acyl intermediate can then go on through further oxidations that are very similar to those that we see in the beta oxidation that occurs in mitochondria. Now, in plants and yeast, acetyl-CoA units can enter something called the glyoxylate cycle. This is similar to the citric acid cycle in some ways, as it involves similar enzymes. However, there's one main difference. We're not going to do any decarboxylations. We're going to start with acetyl-CoA and oxaloacetate using the enzyme citrate synthase to form citrate. We're then going to do the molecular rearrangement using a conatase that we saw in the TCA cycle. But this is where the steps differ. Once we get to isocitrate, we're going to use an enzyme called isocitrate lyase that breaks apart isocitrate to form succinate and glyoxylate. Now, succinate, as you'll recall, is a 4-carbon molecule. Glyoxylate is a 2-carbon molecule. The succinate that's generated can go on to make glucose. And so, in plants and in yeast, we can use acetyl-CoA to form succinate, which can then form glucose. Since mammals do not have the glyoxylate cycle, we cannot use acetyl-CoA to synthesize glucose. Because the carbons that we put into the TCA cycle do not result in a net synthesis of a 4-carbon molecule. Because the 6-carbon molecule that we create is converted into a 5-carbon and then a 4-carbon molecule through oxidative decarboxylation in 2 steps. because of the decarboxylations, there is no net synthesis of a 4-carbon molecule. So now, anyway, since we have glyoxylate, what can we do with that? Well, we can add another 2 carbons through acetyl-CoA, and through the use of malate synthase, make malate again. Malate can be oxidized by NAD+ to form oxaloacetate and NADH, and then we can start the cycle over again. And so again, some organisms do have the glyoxylate cycle, by which they can use the acetyl-CoA that's generated in fatty acid breakdown directly to make glucose. Through the formation of the succinate intermediate, this 4-carbon molecule can then be used to make pyruvate and go on through the gluconeogenic pathway to form glucose. And now we're going to change gears and talk about fatty acid synthesis. Unlike fatty acid breakdown, this occurs in the cytosol. And in mammals, we have one polypeptide that has many enzyme functionalities. This is not just the beta oxidation pathway run in reverse. There are different enzymes, it occurs in a different place, and intermediates are attached to something that's called the acyl carrier protein, not to coenzyme A. The fatty acid chain is going to grow by addition of acetyl units, but we're going to use the donor molecule, malonyl-CoA. The addition of acetyl units is driven by carbon dioxide release from malonyl-CoA. This is entropically favorable and helps to drive the reaction. Now, the maximum length of fatty acid that fatty acid synthase can catalyze is palmitate, a 16-carbon fatty acid. Any further acyl additions or any double bond formations require other enzymes that are found in the endoplasmic reticulum. Fatty acid synthesis occurs in the cytoplasm, but one of the things we need for fatty acid synthesis is acetyl-CoA. This cannot pass through the intermitochondrial membrane where it's produced, and so we need to convert acetyl-CoA to citrate in order to move citrate across the intermitochondrial membrane and into the cytoplasm. Once citrate is transported to the cytosol, it can be reconverted to acetyl-CoA and oxaloacetate in the cytosol. Then oxaloacetate can be converted to pyruvate again for re-entry into the mitochondrion. Now, if you'll notice, this cycle that occurs in the cytoplasm not only produces acetyl-CoA, it also produces NADPH, and NADPH is another thing that we need for fatty acid synthesis. Now, if you'll recall, in fatty acid degradation, the progression of reactions is oxidation, hydration, oxidation, thiolysis. A good way to remember this is O-hot. With fatty acid synthesis, we have the reverse of every single one of these reactions. Instead of an oxidation, we have a reduction. Instead of a hydration, we have a dehydration. Instead of the other oxidation, we have another reduction. And instead of a thiolysis, a breaking apart, we have a condensation, bringing together. I would like to draw your attention to the fact that although these steps were outlined going from end to beginning, the actual progression of reactions for fatty acid synthesis is condensation, reduction, dehydration, and reduction again. Now, we said that instead of using coenzyme A as a carrier of acyl groups, we're going to use an acyl carrier protein. This is found in fatty acid synthase itself, and it uses the same functional group, phosphopentathene, as coenzyme A does. The difference between the two is that coenzyme A is a phosphopentathene group and an adenine, whereas with ACP, we have a phosphopentathene group, and it's attached directly to the enzyme itself. Now, the useful feature of the phosphopentathene group is twofold. This functional group is able to make high-energy thioesters to metabolic intermediates, and thus trap the energy that's contained in those intermediates for use later. And it can also swing those intermediates between different functional groups in an enzyme. And so these are the reactions that we're going to do to make fatty acids. We're going to start with condensation. We're going to add a 2-carbon acyl unit to increase the fatty acid chain length by 2 carbons with each round of synthesis. We're then going to reduce the keto group to an alcohol. The third step is a dehydration. We're going to remove water to make a carbon-carbon double bond. Finally, we're going to reduce this double bond to a single bond to form a saturated hydrocarbon. These steps are going to be repeated through 7 rounds to yield a 16-carbon fatty acid palmitate. The palmitate will then be released through a thiolysis function right within the fatty acid synthase itself. And so step 1 is carboxylation of acetyl-CoA to form the 3-carbon malonyl-CoA. The enzyme that carries this out is acetyl-CoA carboxylase, and again the name just makes sense. We're carboxylating acetyl-CoA. This enzyme requires both ATP and biotin. Biotin, you'll remember, is a functional group that carries carbon units, generally as carbon dioxide. Now, it's important to remember that carbon dioxide does not travel in the system just as CO2, because that, let's face it, is a gas. Rather, it is usually found as bicarbonate in the system. And so when I say carbon dioxide, please understand that this is bicarbonate actually. So acetyl-CoA plus ATP plus bicarbonate is going to yield malonyl-CoA plus ADP, an inorganic phosphate, and a proton. So what have we done here? Well, we've harnessed the energy available in ATP in a high-energy malonyl-CoA unit. We can use the energy that we've trapped by releasing carbon dioxide, and in so doing, the entropic value of this reaction will drive the condensation reaction forward. Now, the carboxylation of acetyl-CoA to make malonyl-CoA is the first committed step in fatty acid synthesis. Here we see the protein structure of mammalian fatty acid synthase. This is a dimeric protein that has 6 different enzymatic capabilities, 5 of which are shown here. The malonyl acyltransferase portion, abbreviated MAT, is shown in red. The beta-keto-acyl synthase, abbreviated KS, is shown in orange, and it's centrally located. The beta-keto-acyl ACP reductase, abbreviated KR, is shown in yellow. And the beta-hydroxyacyl ACP dehydratase, abbreviated DH, is shown in light green. And finally, the enoyl ACP reductase portion, abbreviated ER, is shown in dark green. The functionality that is not shown here is the thiolase portion. We're going to load up the malonyl acyltransferase region, first with the acetyl group from acetyl-CoA. And again, we're just transferring from one thiol to another. We're going to transfer this acetyl group to the terminal thiol group of the acyl carrier protein. But now that we have that acetyl group there, we're going to have to move that over so that we can make room for the malonyl group. So we're going to move this acetyl group to the condensation center. This is the beta-keto-acyl synthase region that's shown in orange. There's a cysteine group there that's going to take that acetyl group. And so again, we're just moving from the thiol of the ACP to the thiol of the cysteine in the beta-keto-acyl synthase region. Okay, so now we've opened the acyl carrier protein. The terminal thiol is ready and available to accept the malonyl group from malonyl-CoA. So we're going to transfer the malonyl group from malonyl-CoA to make malonyl ACP. And now we're going to do the condensation reaction. And this happens in the condensation region. we're going to decarboxylate malonyl ACP to provide the energy needed for the condensation reaction. So here comes the malonyl ACP. It is going to become decarboxylated to form another acetyl group. The carbon dioxide is going to go away. And now we're going to condense these two acetyl groups to form a 4-carbon unit. And this happens through the action of beta-ketoacyl synthase. And now we have a 4-carbon unit. It's a beta-ketoacyl group that's attached to the ACP. We're then going to take this and we're going to reduce it. This is the first reduction, and we're going to use NADPH as a source of electrons. The enzyme that does this is beta-ketoacyl ACP reductase. We're going to reduce the ketone to form a hydroxyl group, beta-hydroxyacyl ACP. And now that we have this, we're going to dehydrate it. We're going to remove water. So we're going to send that to the DH region, to the beta-hydroxyacyl ACP dehydratase, to release water and form enoyl ACP. Now we have an enoyl in this region. We're going to reduce that because what we really want is we want another acyl group, this time a 4-carbon acyl group, so we can add another 2 carbons to it and make the chain even longer. So we're going to go in a cycle. We're going to reduce the enoyl, and this happens in the ER region. The enoyl ACP reductase enzyme activity of fatty acid synthase is going to take care of this. This is reduction number 2. We're going to take this enoyl ACP and reduce it to form a 4-carbon saturated hydrocarbon attached to the acyl carrier protein. And again, for this reduction, NADPH is used as a source of electrons. We're then going to transfer these 4-carbon saturated hydrocarbons to the condensing region in order for another condensation to happen. So now we have the elongated acyl group that's attached to the cysteine in the condensing unit. And we're going to bring in a new malonyl group in order to make this longer by 2 carbons, so that we'll have a 6-carbon acyl group that's attached to the acyl carrier protein. And this will go on until we have a 16-carbon fatty acyl group that's attached to the acyl carrier protein. And in order to release this 16-carbon fatty acyl group, we're going to use the thiolase enzyme of fatty acid synthase to release palmitate. And so to recap, first of all, the acetyl unit is transferred from coenzyme A to the acyl carrier protein of fatty acid synthase. And then it is transferred again to a cysteine in the condensing region. Then a malonyl unit is going to be transferred from coenzyme A to the acyl carrier protein. These events happen through the action of the malonyl acyl transferase, abbreviated MAT. Then comes the condensation. Malonyl is going to be decarboxylated, and the condensation is going to result in a 4-carbon beta-ketoacyl ACP. This is a 4-carbon unit that's attached to the acyl carrier protein. This happens through the action of the enzymatic activity beta-ketoacyl synthase, abbreviated KS. Next comes the first reduction. We're going to reduce the beta-ketone in beta-ketoacyl ACP to result in a beta-hydroxyacyl ACP. This happens through the action of the beta-ketoacyl ACP reductase, enzymatic activity of fatty acid synthase. This is abbreviated KR. And again, we're going to use NADPH as a source of electrons for this reduction. Then comes a dehydration to introduce a double bond. This happens through the action of the beta-hydroxyacyl ACP dehydratase. We're going from beta-hydroxyacyl ACP to an enoyl ACP with a double bond between carbon number 2 and 3. Then comes the second reduction to get rid of that double bond and to have a 4-carbon saturated acyl ACP result. Again, we're going to use NADPH as a source of electrons, and this happens through the action of the enol ACP reductase, enzymatic activity of fatty acid synthase. This is abbreviated ER. And now we're ready for another round of condensation to start all over again with the cycle. And finally, the thiolase activity of fatty acid synthase releases the 16-carbon palmitate from the acyl carrier protein. And so the product of fatty acid synthase is the 16-carbon fatty acid palmitate. Any other features of fatty acids, such as increased length or desaturation, happens through the activity of other enzymes. Regulation of fatty acid synthesis occurs through the regulation of the first committed step that is catalyzed by acetyl-CoA carboxylase. This enzyme is positively regulated by citrate and negatively regulated by AMP through the AMP-dependent protein kinase, and also through negative feedback by the final product, palmitate. Let's look at this a little more closely. Now in this micrograph to the right, we see acetyl-CoA carboxylase as it exists in its active form. You can see that this form is a fiber. And what happens is that as acetyl-CoA carboxylase is activated, it goes from a monomer to a polymer, essentially a fibril. Acetyl-CoA carboxylase catalyzes the committed step in fatty acid synthesis, as we said, and it's highly regulated by phosphorylation and also by allosteric regulation. Now, phosphorylation inhibits acetyl-CoA carboxylase fiber assembly, and if acetyl-CoA carboxylase cannot form the fiber, it will not be active. Fatty acids aren't synthesized when energy is low. We don't want to store fuel if we need energy. And so, when energy is low, this is evidenced by high molar concentrations of AMP in the cell. This activates AMP-activated kinase, which phosphorylates acetyl-CoA carboxylase. This results in inactivation of the carboxylase. It promotes disassembly of the acetyl-CoA carboxylase fibrils into monomers. Now, glucagon and epinephrine reinforce this inhibition by AMP-activated protein kinase by preventing dephosphorylation. Glucagon and epinephrine help to keep acetyl-CoA carboxylase in the phosphorylated state and thus inactive. However, insulin has the opposite effect. Insulin activates the phosphatase, and in so doing, it stimulates acetyl-CoA carboxylase fibril assembly and activation. Glucagon and epinephrine, you recall, are released when energy is low or when you need a lot of energy to get away from something that's really scary. These two hormones do not signal that it's time to store fuel for later. You need it now. Insulin, on the other hand, signals the body that there's a lot of glucose in the system, and it's time to store fuel for later. Now, citrate can bind to and increase the activity of both phosphorylated and non-phosphorylated acetyl-CoA carboxylase by promoting fiber assembly. So even when acetyl-CoA carboxylase is phosphorylated, citrate can cause an allosteric conformational change in acetyl-CoA carboxylase to allow it to form fibrils. Now, it's true that this acetyl-CoA carboxylase is not going to be fully active as it would be if it were dephosphorylated, but it is still able to function. Now, when acetyl-CoA and oxaloacetate are both high, citrate is made, and when citrate levels are high, it's transported out of the mitochondrion. And so what this indicates is the TCA cycle is not going full speed, so there's enough energy around. So there are enough building blocks, there is enough energy. So acetyl-CoA and oxaloacetate will combine to form citrate, which is then transported into the cytosol. And acetyl-CoA and NADPH are made when cytosolic citrate levels are high. However, palmitate can inhibit acetyl-CoA carboxylase. It can also inhibit the export of citrate from the mitochondrial matrix to the cytoplasm. So we get product inhibition of fatty acid synthesis by two means. First of all, palmitate can promote carboxylate fibril disassembly, and it can also inhibit the export of citrate from the mitochondrial matrix to the cytoplasm, and so this allosteric regulator is no longer going to be present. Additionally, we will not have citrate available for the formation of acetyl-CoA and NADPH for fatty acid synthesis. Here again is a table of some naturally occurring fatty acids that are found in animals. I would like to draw your attention to two of them, linoleate and linolenate. The reason for this is because these are actually essential fatty acids. We cannot make these particular fatty acids ourselves, and we must take them in through our diet. And the reason is that we cannot introduce double bonds beyond delta 9. We just do not have the enzymes to be able to do that. Linoleate can be used to make arachidonate. Arachidonate is a 20-carbon fatty acid that is used to make prostaglandins that are part of the body's response to inflammation. Another thing I would like you to know is the chemical composition of palmitate. It is a 16-carbon fatty acid that contains no double bonds, and so its notation is 16:0. Acetyl-CoA lies at the intersection of many different synthetic and degradative pathways. We can break down glucose to make pyruvate and then use that to make acetyl-CoA. We can use acetyl-CoA to make citrate, to make fatty acids. We can use acetyl-CoA to make ketone bodies, which we'll talk about in just a minute. We can also use amino acids to make acetyl-CoA, and we can use acetyl-CoA to make citrate, to oxidize it fully, to make carbon dioxide, water, and ATP. We can also use acetyl-CoA to make cholesterol. This cholesterol can be used to make steroids and also to make bile salts for the emulsification of fats in the diet. The fatty acids that we make with acetyl-CoA we can use to make triacylglycerol, echinosoids, really important signaling molecules, and also to make phospholipids to use in our membranes. So in many ways, acetyl-CoA lies at the center of metabolism. Now in a starved state, we are not going to have glucose in the diet. This can happen either when there's not enough carbohydrate in the diet, or when we're not able to bring glucose into the cell, as happens when a person has diabetes. In diabetes, the cells are not able to respond to insulin to bring glucose into the cell, and so even though there's glucose in the bloodstream, the cells are actually starved for glucose. And so in many ways, the diabetic state is very similar to the starved state. Let's talk about what happens when there is insufficient carbohydrate in the system and the body still needs to function. What we're going to do is we're going to use what we can to make what we need. We need to keep the heart going, we need to keep the brain going, because we need to get food. So we're going to take amino acids and fatty acids, and we're going to break them down to form acetyl-CoA. We're then going to use acetyl-CoA to make something called ketone bodies. Now, ketone bodies are small, water-soluble acids that are the preferred fuel of the heart and also the renal cortex. This is a portion of the kidney that is basically, if you think of the kidney as being an orange, the cortex would be the rind. Ketone bodies can also be used as a fuel of last resort for the brain. If the body is starving, the brain can use ketone bodies under starvation conditions. It can be trained to use ketone bodies. It would really prefer not to. So when we're fasting or in persons who have diabetes, what's going to happen is triacylglycerols are going to be broken apart in the cell to glycerol and fatty acids. These are then going to be shunted through the bloodstream to the liver. The liver is going to turn the glycerol into glucose. Additionally, the fatty acids can be broken down to acetyl-CoA through beta oxidation of the fatty acids. We can then make the ketone bodies. Ketone bodies can be broken down again to acetyl-CoA and can enter the citric acid cycle. Now let's look at the big picture. In the fasting state, the body's main goal is to keep going. And so, adipose tissue breaks down triacylglycerols through the activity of hormone-sensitive lipase. Triacylglycerols are catabolized to glycerol and free fatty acids. Glycerol is water-soluble and is transported in the blood to the liver. Once in the liver, it is used to make glucose through the gluconeogenic pathway. Fatty acids must be transported by serum albumin. They are transported in the blood to the liver. Once in the liver, fatty acids are broken down in the cell mitochondrial matrix. Because the levels of NADH are increased from fatty acid breakdown, this downregulates the TCA cycle by inhibiting isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. Additionally, high concentrations of acetyl-CoA inhibit pyruvate dehydrogenase complex and upregulates pyruvate carboxykinase. The effect of these two actions is that oxaloacetate is used to make glucose. Gluconeogenesis is upregulated, and acetyl-CoA is shunted from the TCA cycle to the production of ketone bodies in the mitochondrial matrix of the liver cells. Two acetyl-CoAs are condensed with the release of coenzyme A to make acetoacetyl-CoA. Then a third coenzyme A and water are used to make 3-hydroxy-3-methylglutaryl-CoA, and coenzyme A is released. Following this, one molecule of acetyl-CoA is released to produce acetoacetate. This is one of the three ketone bodies. Now, acetoacetate can be reduced to make D3-hydroxybutyrate, or because it is unstable, acetoacetate can also spontaneously decarboxylate to make acetone. Now, D3-hydroxybutyrate and acetone are the other two ketone bodies, and so the three ketone bodies are acetoacetate, D3-hydroxybutyrate, and acetone. Now, acetoacetate and D3-hydroxybutyrate can be used to make acetyl-CoA. So these two ketone bodies can be transported through the bloodstream to other tissues, where they can be broken down to yield acetyl-CoA for use in the TCA cycle in other cells. Acetone is a highly volatile substance, and it cannot be used to make acetyl-CoA. Rather, it is excreted by breathing out or in the urine. Persons who are making a lot of ketone bodies will have breath that is rather fruity because of the production of acetone and the elimination during respiration. And so the ketone bodies, acetoacetate and D3-hydroxybutyrate, and acetone are produced in the mitochondrial matrix of liver cells. In this diagram, we see how D3-hydroxybutyrate can be oxidized to produce acetoacetate. Acetoacetate can be broken down to make acetyl-CoA, which can then be used in the TCA cycle. And that is shown here. Acetoacetate can combine with succinyl-CoA to form succinate and acetoacetyl-CoA. Then, through the action of thiolase, acetoacetyl-CoA and coenzyme A can be used to make 2 molecules of acetyl-CoA, and these can be shunted to the TCA cycle. When a person is unable to use glucose because they cannot take glucose into the system, and once ketone bodies form in enough concentration, the fact that we have so many acids in the bloodstream causes the blood pH to drop. If this is not taken care of, coma and death can result. The body cannot function well when enzymes are subjected to a lower pH than is optimal. This is the end of Lecture 27. Have a super day.